Okay, so we've been talking about diffusion, and we've been talking about factors that influence the rates of diffusion of various molecules, whether we're talking about water or other substances. And so then we move into a specific discussion about the diffusion of water, which is basically osmosis. And so there's a class exercise that we will have completed, which is on cell membranes and diffusion or osmosis of water. And as we were discussing the diffusion of water across a semi-permeable membrane, or the osmosis of water requires the presence of a semi-permeable membrane that is impermeable to solutes. So in order for osmosis to occur, these are the two things that are required. And osmosis, or the diffusion of water, then occurs spontaneously. So here we go. Which refers to the fact that it's the free energy of water that moves water from one place to another. Alright, so in that exercise we reviewed things like differences in concentration across a selectively permeable membrane that was impermeable to a solute, such as glucose in this diagram, but permeable to water. So this is just a review of that exercise and some of the terms that we talked about kind of at the end that we will we will conduct that exercise, or we will have already conducted it depending on when you watch this. So in pure water we're looking at a concentration of glucose that is basically, there's no glucose, so it's a 0 molar solution of glucose on the outside in this external environment, compared to this artificial cell that's shown internally with a 0.3 molar glucose solution. And so because the concentration of glucose is higher inside this artificial cell, we refer to this environment as hypertonic. And since the solution outside of the cell is lower in concentration compared to inside, then we refer to this solution as hypotonic. And in a hypotonic solution, the concentration of water is actually higher, even though the concentration of solutes is lower. In a hypertonic solution, the concentration of water is actually lower than on the outside. And like any substance, in terms of the definition of diffusion, water moves from an area of high concentration to an area of low concentration. So water is going to be moving into this cell. Now, the question we want to ask then is, what will happen to the cell once water starts to, move inside the cell? Well, we're looking at diagrams here of plant cells, and what's unique about plant cells compared to animal cells is the presence of the cell wall. So which of these scenarios, A, B, or C, represents what just happened in that example? Over here in A, we see that water is moving in, so this must be the hypotonic solution, whereas inside is the hypertonic solution, which means that water is moving in. And as you can see, there's this, because the cell wall is rigid, we're looking at cell wall right here, and the plasma membrane is this internal area, this internal layer here, the cell membrane. Because of the rigid cell wall, the plasma membrane, even though it's going to be pushing out onto the cell wall, is limited in how far it can push out because of the rigidity of the cell wall. And so that creates pressure inside the cell, which we refer to as, or we would just call the cell as being turgid. Okay, the pressure that's exerted on the cell wall by the plasma membrane and therefore back onto the plasma membrane from the cell wall is referred to as turgor pressure, and it's a term that we will be returning to. Okay, so that turgor pressure is going to be a positive pressure against the cell wall, and therefore the cell wall against the plasma membrane. In this next example, we're looking at the concentration of a sucrose solution in the external environment, out in the beaker, being higher than in this internal artificial cell environment. So we could stop and ask ourselves which one of the cell is basically going to be which hypotonic or hypertonic. Which one is it going to be, a solution that's hypertonic or hypotonic? So the cell will be hypotonic if you tried to answer that question. And outside where the solution it has a higher solute concentration, we refer to this solution as hypertonic. And remember, the hypotonic has the higher water concentration. That's hypotonic. Hypertonic has a lower water concentration. So moving down a gradient of water concentration, water is going to be moving out of the cell. Okay. Now what is that going to mean in terms of the plant cell? So remember, A was where we described a situation where water was moving into the cell, causing pressure against the cell wall by the plasma membrane, and then back on the plasma membrane, which means that the cell is turgid. And so that's positive pressure against the cell wall. Over in C, we see the water here is moving out of the cell. So that must mean that the internal environment is hypotonic, just like we saw in our last example. Outside, the outside environment is hypertonic, which causes the water to move out of the cell. Now what we see these arrows representing is the movement of the plasma membrane away from the cell wall, basically there, and that is referred to as plasmolysis. Or we could say that the cell is plasmolysed. It's rare that we see in nature plasmolysis. This is kind of an extreme example here in this environment. Perhaps a plant that's exposed to high salinity or drought. And then B, we want to sort of take note of here. This is where we have equal concentrations of solutes on both sides of the membrane. And that's going to allow water to come into the cell at an equal rate that it's leaving the cell, or basically the net diffusion of water. Whoops. Of water equals 0. In this case, we would say that the cell is either flaccid, another term we could use, or is, in terms of plant cells here, incipient plasmolysis. It's different than the plasmolysis that we saw over here in C, where water was moving out of the cell at a greater rate than water moving into the cell. And so this is an example where osmotic equilibrium occurs. Again, the solute concentration is equal between inside and outside. And eventually these two, C and A, are going to reach osmotic equilibrium in theory. Now in living cells that isn't always the case, and we'll talk about why that's true. Alright. Because the pressure exerted on the cell wall during osmotic equilibrium is equal both inside and outside, we would say that the turgor pressure equals, or let me write that again, the turgor pressure equals 0. And we use this unit here, megapascals, which we'll be talking about in greater detail when we get to some future topics. Now our question is following this this whole idea that water is moving down its own concentration gradient. We want to really dissect this this whole process, and so the question we want to ask is, why does the concentration gradient of, gradient of solutes, the substances that are dissolved in water, influence the direction of water movement during osmosis? Or we can just say, for short, of why does it influence the direction of osmosis? Is it simply a difference in concentration, or is there something more? What is it about the concentration of water versus the concentration of solutes? Well, the theme that we're going to be sort of playing into in more detail here is that it's not so much the concentration, or let's say the concentration impacts what really is happening, which is that water moves down, not just a concentration gradient, but moves down an energy gradient. And by that we're referring to free molecular energy. So that, again, that energy gradient is from high to low energy, and we'll abbreviate energy with the E there, from a region of high energy to a region of low energy. And that energy that we're talking about, the E, or here we go, E, refers specifically to the potential energy of water. So water moves from an area where there's high potential energy to an area where there's low potential energy. This potential energy that we're referring to, and we'll write this out so we're really clear, the potential energy of water or any molecule is referred to as that molecule's chemical potential. Which we symbolize with this symbol here, mu. So we'll be looking at the chemical potential or mu of substances, or basically of water, and how the potential energy of water is influenced by the presence of solutes and their potential energy. So we'll pick up from here as we continue on with the next video clip specifically on chemical potential of water.